So I will talk about ion channels and transporters in immunity, some known stuff, work that we have done over many years on Orai-STIM crack channels, and then some efforts to identify new ion channels in the immune system. So this is the Orai channel, which encodes the crack channel. It's the main calcium influx pathway in non-excitable cells and also in some excitable cells. Obviously, there are other calcium channels too, but we think that this is a really important one. This gets stimulated downstream of the engagement of immunoreceptors like the T-cell receptor, but also G-protein coupled receptors and others. What they have in common is the production of IP3, which then releases calcium from ER calcium stores through IP3 receptors. And that leads to a transient increase in intracellular calcium, as you know, but it also leads to the dissociation of calcium from the EF and calcium binding domain of this by now well-known molecule STIM1, which is present in a folded inactive state when the calcium stores are full, but once the stores are depleted by IP3-induced store depletion, the molecule undergoes a very strong conformational change, and that allows it to bridge the gap between the ER membrane and the plasma membrane and then bind to Orai-1 and activate the channel and mediate calcium influx. And this calcium influx is called store-operated calcium entry, SOCE. I'm not going to go into any mechanistic detail here. This has been studied in great detail over the last 15 years since we cloned Orai-1 and Micah Halen and others cloned STIM1. So the calcium that enters the cell through SOCE then activates many calcium-dependent enzymes like calcineurin, CAMP kinases that then activate transcription factors like NFAT. And especially in immune cells, NFAT is important for the production of cytokines, chemokines, and as you will see later in my talk, also for the metabolic regulation, metabolic adaptation of cells once they become activated. Just as a bit of background, this is the channel structure that was solved by Shou-Wei Hu in the lab of Steve Long here at Memorial Sloan Kettering in New York. The channel is a hexamer. These are the M1 domains that coordinate the binding of calcium in the pore. This is the pore structure. It's a very narrow pore that a friend and colleague of mine, Murali Parkia, who you hear from later, has really very nicely characterized besides the structure by mutagenizing all these residues and then studying the function of the channel. And the reason I'm showing that is because when we cloned the channel in 2005 and 6, we did this by, among other things, using patients that had a defect in calcium influx, as you can see here after stimulation, and they had a defect in crack channel currents. And we didn't know at the time what the molecule was, right? So we then used positional cloning in patients, a large family that had this phenotype, to then find a mutation in a then unknown gene called Orai, which we named Orai-1. And this mutation is in arginine 91 that is mutated to tryptophan, and this tryptophan blocks this already very narrow pore and prevents calcium influx and crack channel currents. And again, I'm not going to go into detail, but over the years we have identified many mutations in Orai channels, mostly in the transmembrane domain, that don't abolish protein expression but function. And we also identified mutations in the activator STIM1 in patients. Some of them abolish protein expression, but in this case, this R91R 429C mutation, calcium influx is gone, but the protein is here. So what this mutation and others do is they interfere with the binding of STIM1 to the crack channel and thereby inhibit its function. Now, the phenotype that results from mutations, loss of function mutations in STIM1 is this one here. We call this crack channelopathy. So this is certainly an ion channelopathy. This is characterized by combined immunodeficiency. These patients typically die in their first year of life because of overwhelming viral infections. Some of them are associated with tumor development, B-cell lymphoma or Kaposi sarcoma. Patients also have fungal and bacterial infections. They also develop autoimmunity. They have autoantibodies against red blood cells and platelets, and they have non-immunological symptoms, muscular hypotonia that's associated with the type 2 muscle fiber atrophy, and they have what's called ectodermal dysplasia. And I just want to show you 2 slides on that. One phenotype is that these patients cannot sweat, so they have anhidrosis. And we were wondering for a long time, why is that the case? And do they have, for instance, a defect in sweat gland development? But that's not the case. So these are biopsies from patients with Orai-STIM mutations, and they do have sweat glands. But what we realized is, looking at the biopsies, is that the lumen of the sweat glands is smaller. That's quantified here. And we thought that may have to do with impaired sweat production. And when we tried to measure that in mice, we actually found the same. These are conditional knockout mice for Orai-1 and STIM1, STIM2 in ectodermally derived tissues. And if you use iodine starch test and stimulate the mice with acetylcholine, you see that these sweat dots in the paws of wild-type mice do not form in the knockout mice. Mice only sweat in their paws. And so that indicated that crack channel mediated calcium influx is important for sweating. We can measure calcium levels directly in excised sweat glands from the paws of these mice stimulated with acetylcholine. You see this typical FURO2 measurements. Calcium is reduced. And then we thought, well, can we measure currents? What is this due to? So we took human sweat gland cell lines and knocked out Orai-1 by shRNA and stimulated the sweat gland cells with trypsin, which induces a chloride current in wild-type sweat gland cells. And you see there's a very robust current here, but in the Orai-deficient sweat gland cell lines, there's only a transient current that's due to store release, we think, but then there's no sustained chloride current. And then we hypothesized there is likely a calcium-activated chloride current present in these cells that mediates sweat production. And there were two options. One is bestrophin-2. We knocked it out, but there was no effect. And then we looked at anoctamin-1 or TMEM16A. And if you knock that out, then chloride currents are completely gone in these sweat gland cell lines. And so the model that, based on these data and other data we came up with is that store-operated calcium entry leads to calcium influx, and that's required for the activation of the TMEM16A chloride channel. And then secondary to that, you have water efflux through aquaporin 5, which we did not study in great detail. It's expressed, but we didn't do functional studies. So that's probably the explanation why these patients cannot sweat. Another phenotype that these patients have is they have what's called amylogenesis imperfecta. They have very poor dental enamel, which is the hard outer layer of the teeth, as you probably know. And when we had observed many patients, and then we collaborated with a colleague of mine, Rodrigo La Cruz, at the NYU College of Dentistry, and he found that in ameloblasts, which are the dental enamel forming cells, Orai-STIM are actually expressed. And when we take, again, the STIM1, STIM2, CD14 Cre knockout mice, calcium influx is completely abolished in these ameloblasts. And when he then looks at the teeth of these mice, you see that Orai-1 knockout mice have these chalky white teeth, which is kind of a little bit paradoxical, that if you have less calcium, the teeth become white, but that's how it is. And if you do a backscatter electron microscopy, you find that the enamel is actually not formed properly, and the outer layer is very brittle, and that's the reason why it gets used up. And these patients actually have only this underlying dentin layer that is yellow left, and the dental enamel is completely gone. So this means that crack channels are important for tooth formation in both mice and human. But as I said, the main phenotype in these patients is the immunodeficiency and the bacterial infections that they get. And over the last 15 years or so, we worked this out and we looked at different types of immune cells and different T cell subsets. And what we think is that the main reason for the immunodeficiency is the T cell defect in these patients and the role of crack channels in T cells. And when you look at the T cell functionality, you find that T cell proliferation is strongly reduced, cytokine production is reduced, cytotoxicity, basically the ability of CD8 T cells and NK cells to kill virus-infected cells or tumor cells is reduced. And there's also a defect in the production of antigen-specific antibodies, which I'm not going to go into. This has to do with these cells, T follicular helper cells for the immunologists in the audience, which require crack channels for their development. So the proliferation defect was one of the first defects that we observed, and I just want to show you two slides why we think that is the case. So we took a patient with a STIM1 mutation that does not abolish protein expression but calcium influx, and then we stimulated these cells and looked at proliferation. And you can do this with a dye called CFSE. You can basically measure the dilution of the dye with each cell division. In wild-type cells, after stimulation, you see that the initial high peak here moves to the left, right? That means cell division, but it does not happen in the STIM1 deficient cells, right? This means the cells do not proliferate at all, and you can mimic this by using an inhibitor of the calcineurin, which is downstream of the calcium influx. It's called FK506 or tacrolimus, which is kind of the main go-to drug to prevent transplant rejection. If you give patients a new kidney or heart and they're not haploidentical, they're not matched perfectly, you have to immunosuppress them, and you use this drug for that. So crack channels in FK506 have very similar mechanisms of function, as you will see. What we also realized is that when you stimulate wild-type cells, they increase their cell size dramatically. Naive T cells are very small, they sit around. Once you activate them, they blast, they proliferate, and they grow. And if you don't have functional STIM1, or if you block calcineurin with FK506, that cell growth doesn't happen. And we thought about this, why is that the case? And then eventually we settled on studying immune metabolism, and this is just one vignette of data that we published about 5 years ago. This is a polar metabolite analysis using LCMS, liquid chromatography mass spectrometry, where we compare the metabolites in wild-type T cells and STIM1, STIM2 deficient T cells that were either left unstimulated, naive, or stimulated. And you can see that after stimulation of wild-type cells, the abundance of glycolytic metabolites, TCA cycle metabolites, and amino acids goes up dramatically, basically indicating that they undergo a metabolic change in response to the stimulation. This is what immune cells do, and that's what they need to actually grow and proliferate. But you also see that in the STIM1, STIM2 deficient T cells, this metabolic switch is actually much blunted, right? So they don't have glycolytic or TCA cycle intermediates. And without going into great detail here, I just show you the summary of the studies where we are convinced that calcium influx through crack channels is absolutely required for the activation of calcineurin, because we see very similar effects by blocking calcineurin function with tacrolimus or FK506. And this activates NFAT that drives the expression of transcription factors that then in turn activate or transcribe glucose transporters 1 and 3, which we showed in this paper, and also glycolytic enzymes. And that is required for actually driving glycolytic metabolism, including the production of amino acids and nucleotides that allow T cells to pass a cell cycle checkpoint, enter the cell cycle, and then grow and expand. And this clonal expansion is basically at the heart of adaptive immune responses. Okay, so just one summary slide. We have, over the last 15 years or so, looked at many different types of T cell responses. These are pro-inflammatory T cells that are at the heart of many autoimmune diseases like inflammatory bowel disease, colitis, multiple sclerosis, rheumatoid arthritis. They secrete pro-inflammatory cytokines indicated here, and they are very sensitive to, or they need a lot of calcium. If you suppress calcium a little bit, then the function of these cells is suppressed. And but other cells, like regulatory T cells that maintain immune tolerance, that prevent autoimmunity, or cells that are important for antiviral immunity, like CD8 T cells or TFH cells that help B cells make antibodies, right? If you don't have these cells, that would collapse your anti-COVID immune response, for instance, right? These cells are much less sensitive to reductions in calcium. They only require fairly small calcium levels. And the reason we know this is when we use different types of knockout mice that have different levels of calcium deletion, we find that if you only partially delete calcium stop-rated calcium entry, these responses are intact. The Treg responses are intact, but the Th1 and Th17 responses are impaired. And we think that allows for a therapeutic window in which we can use crack channel inhibitors to suppress these pro-inflammatory responses in autoimmune diseases without interfering with these protective immune responses in other contexts. And that is translationally and clinically relevant because a company that I'm involved with, and I should disclose this actually, it's called Calcimedica. I'm a scientific co-founder of that company, has developed crack channel inhibitors, and they have passed, or one of them has passed, 2 phase 2 clinical trials in acute pancreatitis and in COVID-19 pneumonia. And the drug was safe and didn't have apparent side effects, and it reduced the time in the hospital and the severity of disease. So these drugs are now planned to be tested in other disease settings also. So this is actually quite exciting that we come from patients with primary immunodeficiency, crack channel allopathy, identifying the gene and the mutation, to now having drugs that can inhibit this pathway. As I did not mention yet, is so here's the Orai-STIM1 pathway that mediates calcium influx, but there are also other calcium channels. So Barma talked about piezo-1 yesterday. Diane Lipscomb mentioned P2X7 as a potential pathway in the skin, and there are also others. So, but besides calcium channels, there are other ion channels that are reported or have been reported in T cells, magnesium channels, sodium and potassium channels here, and then also an assorted group of other channels. The question was, which of these ion channels are actually really important in T cells? And my lab set out several years ago to study this in more detail in an unbiased, agnostic manner, if you will. The first approach was to just look at expression, gene expression, and we basically curated our own library of ion channels and transporters with 700 plus genes, and then we did a principal component analysis based on all the immune cell expression data from different databases. And this is a principal component analysis, and all you can see here is that B cells and T cells and NK cells are far apart, and that means that they express different groups of ion channels. So they are very they don't all use the same types of ion channels. And we can zoom into this looking at a heat map like this here, where each row is an ion channel or metabolic transporter, and then each column is a group of immune cells, and then their subsets. And you can see that immediately that T cells up here express other ion channels than, for instance, B cells down here, or dendritic cells, or NK cells. And I think that is interesting. Obviously, you can't be deceived by expression, because expression doesn't mean function, but it's a hint. And so we looked at some of these highly expressed ion channels in immune cells. And I want to give you one example of an ion channel that we published this year. It's called LRCHC. It belongs to a family of 5 genes that encode the RAC channel, the volume-regulated anion current or channel, or swell channel. And you will hear more about this from Xiaoshu, I think later today, actually. So this was cloned by Thomas Jensch and Adam Pataputian in 2014. And I'm not going to go into structure. You'll hear more about this in a later talk. I only want to say that this is a swell-activated chloride channel. So if you put cells, T cells or any other cell, into a hypotonic buffer, they will swell because they influx water. And that activates, through mechanisms that are not entirely clear, I think, this chloride current, a chloride channel that's called RAC, volume-regulated anion current, which is actually encoded by the LRRC8 family. And that leads to chloride efflux, followed by water efflux, and that leads to a reduction in cell size. And if you knock out LRCHC and T cells, this regulated volume decrease, as it's called, regulatory volume decrease, is actually impaired. We can, in fact, measure this current directly in T cells by putting them in hypotonic buffer. This was done in collaboration with David Ewell in Rochester. You see a beautiful chloride current here in wild-type T cells, also here, which is completely abolished if you knock out LRCHC. Now, the problem here was that why do T cells need to respond to hypertonicity, right? When do they see this? Maybe in the kidney, but there are not a lot of T cells in the kidney. When would this pathway actually be functionally relevant? And that took us several years to really come to terms with. And eventually we took, but this is relevant in T cells. I should mention this first. Because when we knock out LRCHC, T cell function is changed. The T cells become more functional. There's decreased cell death, so the cells survive better. There's increased proliferation. They make more cytokines, IL-2, interferon gamma. And they also are more able to induce autoimmune disease. This is the EAE model of multiple sclerosis. If you knock out LRCHC, the mice get more sick. They have more CNS inflammation. So clearly, this is a negative regulatory pathway. But why? And we took, and again, I'm not showing you data. I'm just showing you the summary cartoon. We took cues from Lingjun Li's lab at Stanford and others that showed that LRC8A is actually an uptake channel for cGAMP. cGAMP is a second messenger. It's a dinucleotide, cyclic dinucleotide, that is produced in innate immune cells by an enzyme that's called cGAS. And cGAS responds to intracellular DNA from viruses and other sources. But CGAM can also be taken up through channels. And in T cells, that is LRCHC. If you knock it out, the CGAM uptake is impaired. CGAM will activate a sensor called STING, which is in the ER. And STING typically induces a type 1 interferon response. It's the first immediate mechanism, antiviral immune mechanism. Now in T cells, that was actually not the case, as we found, but we found that STING activates p53, and p53, many of you know, is a tumor suppressor. It's kind of a key for preventing cancer growth by imposing cell cycle arrest and apoptosis. Now, when we knock out LRCHC, this p53 levels go down, so we have less cell cycle arrest cells proliferate and they die less. So that's why we have this enhanced T cell functions, and we can test that by knocking out or manipulating different parts of this pathway. So that was a very interesting and I think novel mechanism for this channel in T cells. I'm not sure how much time I have, Rajesh. 2 minutes? Okay, good. Okay, so we are also, besides looking at expression, we are also doing forward genetic screens, CRISPR or shRNA screens. The general idea is that we use our library of hundreds of ion channels, knock them out in T cells, put these T cells back in mice, and then we induce different types of immune responses. And this can be either infection with virus, that can be tumors, or autoimmunity. And then several weeks later, we take out the T cells from the mice, look at the representation of the ion channels in these cells that we isolated from the end organs. And we're doing this in a number of contexts: LCMV infection, flu infection, multiple sclerosis, IBD, tumors. In vitro, we are doing this in macrophages, M1, M2 polarization, B cell differentiation. And there are posters from our lab that you can look at. The ideas, or the hope, is that we can identify ion channels that regulate different types of immune responses. What should I show you? Because, let me skip this. But we had actually done one screen in the LCMV setting, and we found, our postdoc, Axel Concepcion, found several genes, about 14 hits. And one of these hits that we confirmed was actually beta subunit of a voltage-gated calcium channel. And I put this in this morning because we heard a lot about voltage-gated calcium channels. And I wanted to just mention that there has been a history of voltage studies and papers over the last 20 years on voltage-gated calcium channels in T cells. And we were curious, does the fact that we found CAV beta 1 in the screen mean that these are really important in T cells? Nonetheless, we found that when we knock this out, yes, there is some increase in apoptosis and depletion of T cells, but there's really no effect on cytokine production or calcium levels. And that made us curious. And this, we went back to some old data generated with Murali Prakriya, where he measured currents, voltage-gated calcium currents, and calcium influx in response to depolarization. But there's really nothing. And over the last years, we have tried many different protocols, mouse and human cells, under various stimulation conditions, polarization conditions. There's absolutely zero evidence for functional voltage-gated calcium channels in T cells. And I don't know where these papers are coming from and where the effects are coming from, but it has nothing to do with calcium influx, in my opinion. And the reason for that is there are these channels expressed. You can see that by RNA sequencing in human and in mouse, for instance, CAV 3.3 in human T cells. But when you look more closely, which you can do by RNA sequencing, and look at actually the expression pattern or the expression, the transcription of each exon in this gene, what we found for all of these genes that are apparently expressed is that they are lacking the first 10 or 12 exons compared to expression in the brain. And the reason for that, we think, is that there are alternative transcriptional start sites that actually truncate the transcript at the 5' to make a shorter transcript. And if that were actually transcribed and be stable as a protein, that would lack the first subunit and part of the second subunit. So most likely not make a functional channel. And there was another paper that showed the same for voltage-gated sodium channels in a different context. So this may be a more common mechanism why some of these channels are not functional. So with that, I thank several people: Axel, who did the story on LRCHC, Martin and Marta, who did the work on the metabolism story, and who did I forget? Sarab, who worked with Axel on the voltage-gated calcium channels. So thank you very much.
Thanks very much. Time for some questions. Yes.
Great talk. Thank you.
I didn't see you yet. Here.
So if the calcium channels are not functional due to these splice variants, why do you think the beta subunit showed up in the screen? What do you think may be happening? A non-canonical function, or?
Yeah, non-canonical functions. There are actually very long reviews written about this, right? These beta subunits are soluble proteins, and they have been implicated in a lot of different things, in the nucleus transcription, in all sorts of regulation, right? There are probably dozens of alternative functions published for these beta subunits. That's the best I can say. I really don't know what they do.
Yeah, beautiful talk, Stefan. So, yeah, sticking with the calcium channels that are expressed there. So one of the things that I know is that some small G proteins like GEM are expressed in immune cells. GEM is a small G protein that inhibits voltage-gated calcium channels. And when Steve Marks talks, we realize that when these RGKs are present, the channels are constitutively inhibited, but they can be relieved of inhibition by phosphorylation. So just wondered whether something like that could be at play. And you could have these channels that are functional, kept in a basally inactivated state, but can be turned on by a second signal beyond voltage.
Yeah, I mean, it's like chasing the Flying Dutchman or something. It's chasing a ghost. I'm happy to send you T cells. I'm done with this. You know, I decided this is not going anywhere. No, it's a good point. I agree with that. You can study these mechanisms, but we tried so many things. I don't think there is anything. Murali, go ahead. You can answer your own question, by the way, because if you ask about the last part.
This is related to the metabolism part of your story, and I was actually wondering, it's common, it seems like calcium drives ETC and OXPHOS, and you showed evidence for that in T cells. But since you have a pretty substantial reduction in glycolytic enzyme expression and byproducts, pyruvate, that feed into the ETC, I was wondering, is the primary effect on metabolism, in fact, an effect on glycolysis rather than oxidative phosphorylation? And is that just purely a consequence of the loss of the metabolites that feed into the ETC pathway, or the loss of the glycolytic enzymes that would have the same effect?
Yeah, I mean, I think the fact that you don't have the glycolytic metabolites that feed the TCA cycle, and then also the mitochondrial respiration, right? That is certainly, I would guess that is contributing. But as you also know, I think, is that we have additional data that in Th17 cells in particular, that genes of the electron transport chain are broadly reduced, right? It's like there are dozens of these genes that are also encoded in the nucleus are reduced. So even if you could do the experiment where you reconstitute some of the maybe glycolytic metabolites and then see if that can rescue. My guess would be it cannot rescue mitochondrial respiration, because that machinery is also affected. Right.
Okay, thank you so much.